Hi everyone, my name is Annika Faucon and I'm a PhD candidate at Vanderbilt's Genetics Institute. My research in human genetics focuses on complex polygenic risk. My talk today is going to focus on the Julia package that I've created to generate polygenic risk scores called PRS.jl in collaboration with Julia and Samaru, and it's supported as part of my research in the Cox and Davis labs at Vanderbilt University. Genetics is a field that's tightly intertwined with computational and statistical sciences, but even so, many of our programs are written by people like me, people who aren't doing a PhD in computer science, but want to make a meaningful impact on the field. Biology and related fields have often used Python and R, which have a variety of efficient libraries, but writing an efficient library in them often requires using a second language like C++. Julia solves this two-language problem and makes iteratively improving such libraries feasible for us scientists. Additionally, by improving efficiency, you democratize science. Human genomics is overrepresenting wealthier countries like the US, Australia, and European nations, which tend to have more powerful computational resources and greater funding for the sciences. By reducing computational needs of key algorithms, less wealthy nations can utilize these algorithms to benefit their populations as well. Knowing the impact and the utility of polygenic risk scores and wanting to drive development in polygenic risk score calculation and application, the NIH recently funded a large initiative to develop packages like PRS.jl and their applications for diverse populations. So first, what are polygenic risk scores? Most traits have a polygenic architecture, meaning that rather than having just one gene that causes disease, they have a lot of variants all over the genome that have a really tiny effect. And all of us have some protective variants and some risk variants, but some people have more of one type than another, putting them at a higher or lower risk than the majority of the population. For example, in coronary artery disease, if you look at the tails of the distribution, you can find individuals at threefold, fourfold, even fivefold more risk genetically than the rest of the population. As a result, scientists and clinicians have suggested using polygenic risk scores to help determine 10-year risk for heart attack in the context of deciding if it's time to put someone on a statin. In addition to guiding timing for therapeutic intervention, polygenic risk scores have been studied in the context of determining genetic susceptibility to and predicted progression of disease, as well as therapy response. So how do we get these scores? So we start by calculating the association between a disease and each variant in the genome. So we call this a genome-wide association study. And the result can look like this plot here on the right. So here the millions of genetic variants that are tested for their association with disease are in order of the genome, so across all chromosomes, and each little dot is a single genetic variant. To get a polygenic risk score, most naively, we just create a linear model with an individual's genotype at each of these millions of locations in the genome as input, and use the weights calculated in the association test applied to it, and output a risk score for the disease under consideration. One thing that you might notice is that most of these genetic variants have a significant association with the disease, have a tower with a lot of other variants together too. The thing is that genetic variants don't travel alone. They travel together with the genetic variants that are closest to them between recombination hotspots, which is a phenomenon that causes variants to show a greater effect due to non-independence between variants in the same location. One of the big areas of development with polygenic risk scores is how to deal with this redundant signal. Trying to overcome this complication has given rise to many different kinds of algorithms. One popular approach used by the PRECISE package is to use only top signals and to discard all other signals as redundant signal. However, the problem with this approach is that some true signals may be eliminated. So an alternative approach to throwing out redundant signals is based on the Bayesian continuous shrinkage prior. Simplistically, this approach tries to keep all signals, but gain a clearer picture by shrinking out local signal redundancy. In the PRS methodology, an MCMC algorithm is used with a continuous shrinkage prior to generate higher accuracy models, although it's an inherently stochastic approach. Regardless, for a given amount of computational runtime, PRS generates a higher accuracy model than PRECISE. The original PRS algorithm is implemented in Python, and as a frequent user of this package, I was sometimes limited by the computational overhead. So we were determined to see if we could get better performance by porting the code over to Julia. We figured that Julia's highly optimized matrix and broadcasting operations, together with a real type system, would allow us to achieve a larger speedup. Once we ported the code and saw that it worked on test data, we had to validate that it produced approximately the same results as the original PRS.CS package. Because the approach is stochastic and the random number generators for the MC and C processes are the same, the correlation wouldn't be expected to be completely the same, but we can see that the correlation of scores calculated for people is quite close, with correlations above 0.99. The figure on the slide shows PRS scores calculated for individuals in the Vanderbilt Biobank. As you can see, the scores for this trait are quite similar when calculated with Python or Julia. So next, we wanted to evaluate changes to calculation time on the same CPU architecture. So I was happy to find that the Julia port was anywhere from 4 to 10 times faster than the Python code, averaging about 5 times speed increase. In addition to the speed increases, we made a few improvements around usability and robustness. So first, the columns of the summary statistics file can be given in any order. The summary statistics file can use any delimiter. The missing value indicator can be specified. In Julia, also CSV reading can use multiple threads. And also in our package, you can specify multiple specific chromosomes to use at once. So future directions. Going forward, we have a number of ideas for ways to improve PRS.jl, and we really hope that the community will be able to help. One area for performance improvement that would be using GPUs to compute the PRS model. This could be used throughout the MCMC algorithm to greatly improve performance, especially for calculating large models. Next, we'd like to generalize data loading and saving mechanisms so that arbitrary table types can be used as input and output, as well as internally for performance benefits. This could allow the usage of alternative data storage formats like databases and would make it easy to use PRS.jl within a larger Julia pipeline. Additionally, we'd like to research the feasibility of swapping out the MCMC algorithm for another related stochastic approach, as well as integrating other PRS methodologies. And finally, we'd like to implement the transethnic functionality found in PRS-CSX, which is an extension of PRS-CS that provides accurate generation of models on multi-ethnic genetic input data. I'd like to thank my labs and committee for their support, as well as Julian Samery with the MIT Julia Lab for collaborating with me on this project, and Tianji at MassGen for providing guidance. That's all I've got for this talk. Thank you so much for listening.